Beautiful.
Okay, so thank you for the introduction and for the opportunity to present my work here. So I'm going to talk about today how to examples for translating insight in neuroscience to machine learning algorithms that are better at continual learning. So first, what is continual learning? So to better understand that, I'm going to compare it with conventional learning. That is the type of machine learning we're most familiar with. So you start with a designated problem. For example, you're building a face detection model, and then you collect all the data you can, and you feed it into this model, and then you train it. And once you train it, the parameters in the model stay fixed, and you deploy it for further use. So that's why it's called static learning, because after you train it, you don't change the parameters anymore. You don't do any additional training. And also, you can see how that makes it difficult for a single model to perform multiple tasks. So basically, one model in this paradigm of training can only perform a single task. That is in comparison to our continual learning, where instead of collecting all the data and train them at once, the data comes in continually and the training happens continually, one task at a time. So for a single model, you are able to perform multiple tasks. And you can see that the major difference between continual learning and static learning is that the training data are fed into the algorithms one task at a time instead of just collectively at the same time. And also, in order to achieve this, the continual learning algorithms need to fulfill two criteria. They need to be adaptable and also stable. What this means is that you need to be able to learn new data fairly quickly, and also at the same time retain the information you have already learned. And of course, you will also want other like factors for continual learning as well, such as feed-forward learning and feed-backward learning, which means you want to use what you have learned to help you learn new things. And also you want to, like after you get some new information, you want to go back, and if there's a mistake in the memory, you want to go back and fix that mistake.
No, we can't.
Is there an echo? Anything?
I mean, this is the only, they want to see, I don't know what they want, but just continue.
Okay. All right, so there are many potential applications for continual learning. For example, in AI healthcare, you need to use continual learning models to basically design the treatment for patients depending on the stage of treatment and how far the process is made for the patient. And also for self-driving, where you want the system to adjust to neural conditions based on what you have trained and to adjust to different weather conditions. It's going to be a very beneficial factor if they can achieve continual learning in self-driving systems, and also for personalized AI assistant and so on and so forth.
Oh, question.
Yeah.
So, continual learning, is just, could you conceptualize it, is this process in which a machine learning agent or algorithm is doing one task and getting new data all the time, and you want to incorporate that data? And is this distinct then from the scenario in which you would want a machine learning algorithm or network to learn new tasks using the same data?
You need to learn new tasks, not necessarily with the same data. You can incorporate new data and learn new tasks.
Okay.
Yeah.
So both of those scenarios are in the
Right.
what you touched on. Okay.
Yeah. So in the title, I said I'm going to explore continual learning mechanisms from the olfactory circuit in Drosophila. This might seem a little bit weird because why olfaction and why Drosophila? So olfaction makes nothing to be a very vital sense for us in modern life, but olfaction is actually a very underestimated sense, and animals do need it to survive in the wild. And the architecture of olfactory circuit is relatively conserved across different species. And this suggests this circuit might be evolutionarily optimized for learning continuously. And also, the odorants, like our perception of the odorants is actually hugely impacted by environmental factors such as humidity and the speed of the wind. For example, if you go into the forest in a sunny day, you will smell very differently compared to a rainy day. So in order to adapt, or in order to survive, basically, animals need to quickly adapt to the environmental changes. And all these features make olfaction a nice model to study continual learning because of the because our animals need this capability to adapt to changing environment and also to maintain the knowledge they learned before. And then why Drosophila? Drosophila is a very good animal model. It's very simple and yet it is able to perform many complicated tasks. And also there are lots of experimental data and studies. And so there are a lot of data techniques to build computational models. And what's more, there are comprehensive genetic toolkits. So if you find something or in the computational world, you can have follow-up studies for experiments. So now let's go into some challenges you might think are related to achieve continual learning and what we can learn from the class. So the first challenge is called catastrophic forgetting. And what is that? Let's again look at an example. So suppose I'm building a model to classify the 10 handwritten digits, and I collected all the data and this data set, and I'm going to input all the data at once to the model. And based on nowadays our machine learning algorithms, we can get a near human accuracy for this test. However, if I just make a tiny change to this training process, as I described before, instead of input all the data at once, I have trained them to learn one task at a time. For example, I ask the model to learn 0 and 1 first, and then 2 and 3, and so on and so forth. Even if I'm going to input them into the same classifier, after I trained with task 5, and then if I go back to test the model's performance on task 1, I will get near random negatives. So this is the catastrophic forgetting, which means the model forgets about what it learned before. And why does this happen? So if I'm just going to put all classes together for training, I will end up with a loss function with a very complex landscape. But as complex as it is, our modern day optimization algorithms is able to find its global minimum fairly easily. And you can get a good classification results based on optimizing the function. However, if I'm going to do this one task at a time, I will end up with a sequence of easy but of simpler loss functions. However, by the time when I converge to the global minimum in task 3, there's no guarantee where I will be regarding to the loss function of task 1. So that's why when I'm done with task 3 and go back to task 1, I might get a very bad classification performance. Now we know that flies are able to learn to associate odors with different behaviors, like they are trying to approach some odors and avoid some odors. And also they are able to retain this memory for a fairly long time period. So how do flies do that? And also, can we learn some insights from this? And to tackle this problem, I'm going to ask 2 questions. First is how do flies represent odors in their brain? And the second question is how do flies store this learned odor behavior association? Now let's look at the first question. After some pre-processing in the nose of the fly, each odor is represented by a vector in the 50-dimensional space using a layer of neurons called projection neurons, or PNs here. And now, there will be an extension in dimensionality from here, from 50 projection neurons to around 2,000 canyon cells, which is kind of the analog of brain to the fly. And the projection between the projection neurons to the canyon cells is made by a sparse random binary matrix, so that each canyon cells pulls from about 6 randomly selected projection neurons. And after this expansion in dimensionality, here comes another interesting manipulation. So now that the order is represented by a 2000 dimensional vector, and each of them have different activities. And now the 2000 canyon cells will send its activation to a single neuron called APL. APL is a very large neuron in the flight brain. Actually, there's only one APL in each hemisphere of the flight brain. And APL will send back a very strong inhibitory signal to all the canyon cells, so that the end result is only the top 5% neurons that are in the highest will be active after this inhibition. And this process is called winner take all. So you can see here, after this winner take all process, the order is now represented by a very sparse vector in the 2000 dimensional space. And this is called a sparse coding, which is in comparison to before the winner take all, all the canyon cells have some activities for this order, so this is a dense coding. So you can see the order is finally represented in the flight brain, it's presented by this sparse coding. And the benefits of doing that is that it could reduce the interference of different orders. And what's more interesting is that this reduction of interference is not done through any learning, so it's unsupervised. There's no learning required to reduce the interference of different orders in this manner. Now, let's move on to the second question, but how do flies store learned order behavior associations? So let's say we have an order A, now is represented by this vector of canyon cells here. And downstream of canyon cells, there's another layer of neurons called maximum volume output neurons, or M-bounds. There's only about 34 M-bounds in the entire brain of the fly, and you can roughly interpret as each M-bound representing one type of behaviors in the fly. So I'm showing 2 M-bounds here, one for approach behavior and one for avoid behavior. And the canyon cells and M-bounds make all-to-all connections. And I'm highlighting here the connections between the canyon cells activated by this order A and the M-bounds. Now, if order A is a novel order to the fly, it has never experienced it before, the approach M-bound and the avoid M-bound will receive relatively the same amount of activations from the canyon cells, so that our fly will neither approach nor avoid, or it will randomly approach or avoid this order. Now, if you want to train the fly to approach this order, you might add a sugar reward during the time when the order is present, and this will activate another type of neuron called dopaminergic neurons in the fly's brain. And what it does is to release dopamines between the synapses, around the synapses between the active cases and the avoidant, and will reduce the synaptic strengths between those neurons, so that the end result is the amount of activation received by the approach neuron will be larger than the amount of activation received by the avoidance neuron. So the fly will learn to approach this neuron, and vice versa if you want to train the fly to avoid this order. And you can see that this type of learning only engage a few connections, a few synapses, just the synapses between the active canyon cells, activated by the particular order, and also the corresponding output neuron. And this is called associative learning, which is a quite common learning paradigm in the brain. And during this type of learning, only the weights connecting vector neurons and corresponding output neurons are updated, and other weights remain unchanged, so that you can avoid the learned information being overwritten by learning. And Another thing to notice here is that there are no predictions made during this training. So there's no error use, so no error or assignment, no credit assignment or back propagation involved in this type of learning. Now we find two features of learning of olfactory learning in the fly, and we want to basically translate it into an algorithm. So we started with dimensional vector and go through the dimension expansion and linear take-off process so that each input is now represented by a sparse vector in very high dimensional space. And then we modeled the can you see this? Okay. And then we modeled the associative learning, which we call here partial phrasing, because most of the weights during this training are frozen, so unchanged. So we also call this partial phrasing, and we translate it into the mathematical form. And now that we have the algorithms, what we call fly model, we want to test how it does in a continual learning scenario. So the task we have for this is we created a sequence of tasks. So for example, during training, we asked the model to first learn 0 versus 1, and during the test phase to classify the learned digits. And then in task 2, it learns 2 versus 3. And now that it has learned 4 digits in the test phase, it will be asked to classify all 4 digits together, and so on and so forth until it learned all the tasks we have played. And now let's look at the results. So I used here a data set R, which is the combination of MNIST, the 10 handwritten digits, and combined with another benchmark data set, Flash MNIST, which is a grayscale image, which contains 10 categories of grayscale image for our clothes items, like jackets and all that. So there are in total 20 classes in this data set. And you can look at the x-axis, which is the number of classes that have been trained or have been trained so far, and the y-axis is the test accuracy here. And I want to draw your attention to two control models, offline model and vanilla model. So the vanilla model is just a plain feed-forward neural network with no continual learning modifications. So you can see how fast its performance decrease as the number of classes trained, like the increasing number of classes trained. So basically, this is another exemplification of catastrophic forgetting. So if you don't do any continual learning modifications, that's what you will get. You don't gather good accuracies after you train, like for the only after you're train 2 tasks. And on the other hand, the offline model is a model that does not do continual learning. So at each step, I will just feed in all the data together to help it train, just like the conventional machine learning paradigm, just like the static learning. So for example, here I will just input all the data from the 12 classes to this offline model and let it train to classify the 12 classes and have its accuracy. So the offline model are actually the upper bound of the performance for all the models. And also compared the fly model with 3 popular continual learning algorithms, EWC, GM, and VIR. They also are inspired by our biological systems. And we can see that the fly model outperforms all 3 of them. And not just in this data set, but in the second data set, which contains 100 classes of real-life images. And you can still see that our fly model is the best one here. And I've showed you that fly model actually combines 2 features, sparse coding and partial phrasing. Now, are the 2 features equally important? Can we just achieve this performance using just one of them? So the short answer is no, we cannot. You do need both of them. And I tear apart the sparse coding and partial phrasing and pair it with with other components. And you can see that it's only using partial phrasing without sparse.
Keep going.
if you're only using partial phrasing without dense coding, there's the performance is really bad. And on the other hand, using sparse coding was our conventional like our that for our learning, you will get you will get okay response, but still much worse than combined sparse coding and partial phrasing together. And this is also true in the second data set.
But you expected that when you increase the number of classes, improve the accuracy, right? I don't know. I don't understand why when they take more time to learn, the accuracy go down.
It's different classes, so it's not because you learn more. You add more data, and the data and the results is reduced. It's because you learn different classes compared to before, and after you learn new classes, you forget about what you learned. So as you learn more and more classes, you forget more things, and you only retain your current understanding. So that's why it deteriorates as you learn more classes.
Okay.
So
Question.
yeah,
How many Kenyan cells do you have?
for the MNIST data, I think that's about 4,000 or something. For the CFAR 100 is that is 20,000.
20,000.
Yeah.
So if you increase the number of Kenyan cells, does the forgetting become slower?
Not really.
Okay.
So I tried a couple more Kenyan cells, but the result doesn't get significantly better. And I also played around with the sparsity level. So the current is kind of the best scenario here.
Okay.
So in summary for this part, I showed a neural algorithm for continual learning following detailed circuit mechanism in the through-fly olfactory system.
So and the before chart is experimental or is theoretical? Is numerical the solution of the two?
This is numerical. So
Yeah.
I'm testing the two empirical data sets. The benchmark data setting are usually used in machine learning field.
Can I ask a question as well?
Sure.
Yeah. So part of your solution is freezing, right? The some of the weights. I don't know if you mentioned it or not, but is that like, does the brain do that? And that's one question, and the other one is if the brain does, or like I'm worried about problems with extinction, right? Because the brain needs to remember and learn things, but it also needs to get rid of memories. Would that be a problem? Yeah, if you could comment on those.
Okay, sure. The first question is, yes, the brain kind of does that. So it's at least in the implied, the only synapses that's being manipulated is between the Kenyan cells that are activated for the odor and the enzymes that is specific for the behavior. So this also ensures the specificity of the learning. So you only respond to this particular odor and not other odors with the designated behavior. And for the second question, I think for memory extinction I think actually, well, we haven't really studied this. I think it's a very good question, and but I think it will actually help with memory extinction because you have a designated basically a designated synapses or the type to change for the memory. So you only need to go back to where the locations you made the changes to undo the changes. So so that you do not harm the other memories when you undo what you want to remove. So that answer your question. And also I'm going to talk a little bit more about the forgetting in the following.
Yeah, yes, thank you.
Okay. So okay, as I was saying, this flight model actually combines two methods, sparse coding and associative learning. And none of those methods are new in machine learning field. Basically, they have been known for a long time. However, here I show that Fly combines them in a novel way to solve this to reduce catastrophic forgetting. So this also shows that nature is able to combine like two very simple ideas in a unique way, so to solve a not so simple problem. And this algorithm is lightweight and it's very easy to implement, and it requires no back propagation. And also, some of you might notice that the Fly model, especially the associative learning or partial breathing, resembles the perceptual algorithm, but there are subtle yet important differences. So if you're interested in that, you can go to our paper for further discussions. And now let's move on to the second challenge, which is our noise filtering. So we know that data cleaning is an important step in data analysis. And some people might even say it's the most important step in data analysis. And the difficulty to do that in the continual learning paradigm is that you need to do that online. So as the data coming in stream, you don't know what is the data coming after this. So you need to continue performing data cleaning while expecting, while you don't know what are the data that's coming. You don't even know what is the noise. And is there something like similar that the Fly does or our brain does actually to achieve this? Well, there is, and so we found that the phenomenon habituation has shares some similar functions here. So habituation is actually a very common phenomenon. We experience it every day. So suppose you walk into a coffee shop in the morning and you immediately smell the strong coffee aromas here. However, after you stay there for a while, for example, you walk in there for 30 minutes or an hour, the coffee smell will be faded into the background. So you're not aware of it anymore. However, if there's a new odor, you are like noticing odor come out, like odor or something is burning, you will be very quickly detect that smell. Even though that smell is a very faint smell compared to the strong smell of coffee in the coffee shop. And especially if you compare to someone who just entered the coffee shop, the person will have a much more difficult time to detect the burning smell from all the smells of the coffee. And what I just described are the three features of habituation. So first, it will decrease the activity to repeated neutral stimuli, aka noise. And also, this reduction of activity is stimulus specific. You only decrease your senses to the background odor that conveys no useful information at the time. But when a new odor comes up, like the burning odor, you will be able to detect it right away. And also, this process is reversible. So the next day, when you go to the same coffee shop again, you will smell the coffee. And the questions I want to ask here is that first, what certain mechanism can fulfill all the three features at once? And second, how does habituation help the detection of subtle but important odor despite the strong background? And the third is how to filter out background noise on the fly. So it turns out that the Fly probably uses a mechanism called negative image model to achieve this. And to understand this model, let's go back to the circuit map of the Fly olfactory circuit. So in the last project, I talked about the right side of this circuit, and now we're going to focus on the left side. So the first layer of the olfactory circuit is a layer of neurons called odor receptor neurons, or ORMs. Those are the neurons directly interact with odor chemicals. And after it exchanges or translates the chemical information to neural information, it will send are activation signals to both the projection neurons and the lateral inhibitory neurons, LNs. So let's look at one of those triangles in detail. So because the lateral neurons, LN and PN, are both one synaptic step away from ORN, they will receive our activation signal about the same time, and they get activated around the same time. And because their activation are about around the same time range, they are even learning that their synaptic strengths will be increased. And because LN is actually inhibitory to PN, some people also call this learning anti-figure learning. And the amount of reduction, the amount of strength or increase in the synaptic strength is proportional to the activation of PNs and LNs. And you can see that as our time goes by, and if you continue experiencing this order, and this order conveys no information, so there's no other changes in your internal state, then the final result is that the synapses between LNs and projection neurons will form an image of the incoming order. And of course, it's encapsulated, so it's an active image. So even though the ORNs can still respond to this order, the activity stops at projection neurons. And because the activities of projection neurons will be inhibited by lateral inhibitory neurons, so your higher brain region will not detect this order anymore. And this is the negative image model. And we've translated this biological insights into a mathematical language here. And so we're interested in how these are manipulations in the prefrontal, in the periphery sensory system could affect how we perceive orders. So now for the simplicity of comparison, I'm going to use a binarized representation for orders in canyon cells. So what that means is I'm only interested in the identity of canyon cells that are that are active for a given order and not their amount of activities. So then the identity of the canyon cells response to a given order will be called the tag of that order. And now let's see how the capitulation can change our perception of orders. So let's say we have a mixture of two orders, order A and order B. And order A is the dominant component here, containing 80% of the signal, and order B only contains about 10%. And however, order A is the background here that we want to filter out, and we want to detect the faint order B here. Now you can see that if there's no habituation or whatsoever, the tag of the mixture will be very close to order A. So you see there's a big overlap between the tag of the mixture and the tag of order A. And the tag of order B is only slightly overlapped with the mixture. And then if you habituate order A before and you form this negative image, so the response to order A will be suppressed. And now you can, when you smell the mixture again, the tag of mixture will be completely overlapped with the tag of order B. So you can easily detect order B from this noisy diagram. And in addition, habituation can also have another benefit, that is to facilitate the discrimination between very similar orders. Now, suppose we have three very similar orders, and their ORN responses are highly correlated, like this. And let's say, if you want to discriminate between order SB and order SC, you might have a very difficult time doing that. Just like when people are trying to discriminate two types of roses, it's very hard to tell them apart just by their smell. However, if you habituate yourself with order SA, and after that, you can see that the responses in the projection neurons for order SB and order SC are now quite different. And if you look at the order tag in the canyon cell level, you see that before habituation, their tags are very similar. There are a lot of overlaps. And after habituation to ORSA, you can easily tell our ORSC and ORSC apart as their tags are completely different. So, in recap for this project, I've developed a learning algorithm for short-term odor habituation based on the negative image mechanisms in fruit fly. And this algorithm demonstrates how habituation can help filter out background noise and facilitate discrimination between similar odors. Obviously, without prior knowledge to what the odor coming in. And also, it can lay out a potential framework for online noise filtering that might be implemented into continual learning scenarios. And now let's move on to the third challenge faced by continual learning, that is regularization and normalization. This might be usually overlooked, but actually normalization and regularization are very important for continual learning. Because again, based on the nature of continual learning, the data is coming in a stream, and you don't know what are the data will be coming in, and they might have very different statistical distribution compared to previous data you have trained. So it's important to normalize data so that they have similar distributions to maintain the robustness of the model. And in the brain, there is a phenomenon called homeostatic plasticity, which aims to stabilize our brain. And this can share some similarities between our normalizations in the machine learning world. And homeostatic plasticity is actually an umbrella term, and one of the mechanisms for homeostatic plasticity is synaptic scaling, and that's probably the most well-studied mechanism for homeostatic plasticity. And what it does is that, suppose I'm a neuron and I receive inputs or activations from my presynaptic partners, and if my activity is determined under active, then synaptic scaling will increase my synaptic strength so that I receive more activations from my presynaptic partners, and vice versa for if I'm determined as an overactive. And in the our machine learning field, or basically in the deep neural networks, there is a famous normalization method called batch normalization. So in the conventional, like a standard network, suppose you have your inputs and you normalize your inputs, and then you feed it into the network and it will be multiplied by the main matrix, and you get another vector y, and after you go through a non-linear activation, probably ReLU, and this will be your input to the next layer of the network. And this input to the next layer is usually not normalized. And what batch norm does is that it inserted a normalization in this gap, so that the input to the following layer is also normalized. So you see it first standardized, and then it learns another mean or another scale. So basically learns a different distribution, and then you insert this normalized input to the following layer. And this this manipulation may seem like a simple one, but it has very powerful results. It can greatly reduce the number of iterations you need to train a model, and also it greatly increases the accuracy of the model. However, why does this work, and what is the mechanism of make batch normalization so powerful is still up for debate. So people are still trying to figure out why a simple manipulation like this is able to perform such good results. And I'm going to show you here that actually batch normalization is functionally equivalent to synaptic plasticity. So first, we see there are similarities between goals of normalization in machine learning and homeostatic plasticity in the brain. So they both attempt to stabilize and bound neural activities in an unsupervised manner. And also, they both aim to increase the learning efficiency. And are demonstrated by decrease the number of iterations it needs to converge. And also they're both temporarily local in the sense that the changes only depend on recent behaviors. And now, as I said, batch normalization and a model inspired by synaptic scaling have some similarities, and I'm going to show you like in a minute. So I'm just first pointing out what are the similarities. So they both improve classification accuracy and both increase coding capacity, defined as the number of unique patterns that can be presented by using only the fixed number of neurons or nodes in the network. So I'm going to demonstrate this with the dataset CR10, which contains 10 classes of real-life images using the base model LeNet5, and those are the parameters. And here you can see the results. So basically, you see both batch normalization and synaptic scaling performs much better than the vanilla model without any inserted normalization. And I mean, even with very few iterations, it already gets a much better performance or much better task accuracy compared to the vanilla model. And the synaptic scaling and batch normalization almost have identical behaviors of performance here. And why could that happen? And also, then I'm going to talk about the coding capacity here. And why could that happen? And one hypothesis is that it might be because they both improve the coding capacity by utilizing each node with similar probability. And what that means is this. So suppose we're looking at the vanilla model, and we're looking at how the nodes in the vanilla model are being used. In particular, the two fully connected layers are in the LeNet5 model. And each script here is a node or a neuron. And the color here stands for the probability of this node being activated throughout the iterations. So the brighter the strip, the more likely the node is going to be used. And each row is a different iteration. So you can see some node may start to be used and then it's not used again after some iterations. And you can see a picture here is that you see a lot of strips. That means there are also a lot of black and empty spaces here. Means those neurons almost never get used throughout the training. And you can see how this might not be good for because you can see how this reduces the coding capacity by not using some neurons at all. Right. So then if you add in batch normalization, you see that the neurons are being used more homogeneously, and each neuron have a similar probability of being activated throughout the training. And the same thing happens if you use synaptic scaling. So to paraphrase, what this means is that the batch normalization and also synaptic scaling can increase the coding capacity by utilizing each of these neurons with similar probability and increase the number of information or number of data points it can code it into this network. And this you can see in the vanilla model, you see the scripts, and you might think it's a very sparse usage. But this is very different from the sparse coding I talked about before, because in sparse coding, even though only a few neurons active for a single order, but each neuron have similar probabilities being activated by the orders. However, in this case, some neurons are never being used, so which really reduces the number of orders you can represent. So you'll waste a lot of dimensions even though you have a large dimensionality. And homostatic plasticity does not only happen in one level, actually it happens levels in the neural system, like single neuron levels, networks, layer of neurons, and network of neurons. And for each level, there are some like corresponding algorithms being developed in the machine learning field. For example, batch normalization, group normalization for single neurons, split normalization, layer normalization, and so on and so forth. However, there's still no, as not to my knowledge, no normalization methods for a network of neurons, and that might be interesting to look at, or maybe we can combine different normalization to see if it makes a better performance for the network. Now, the summary for this part is that the normalization method in deep learning are functionally equivalent to homeostatic assistive mechanisms in the brain. And that shows you a toy model inspired by synaptic scaling that could achieve similar effects of batch normalization, at least for this simple classification task. And also, the regularization mechanisms in the brain could inspire more normalization methods for continual learning algorithms or machine learning in general. So, future direction I've talked about how important it is to avoid catastrophic forgetting, but in fact, forgetting is a very healthy part of learning because there are limited capacities of the network. So we need to basically manage the memory to carefully allocate our space for new tasks and old tasks. And also we need to be able to go back and fix the old memories that might have some mistakes. And to do this, we need to forget gracefully, which means you want to forgetting while keeping the system and other memories intact. And there have been some demonstrations for graceful forgetting in Drosophila, such as memory extinction and transient forgetting. So I would be interested to look at those mechanisms and try to integrate them into the continual learning algorithms. And another direction is that how to make a robust decision, because if you're like continual learning a lot of tasks, there must be some conflicts between the different tasks. So you need to make decision in a context-dependent manner. And how is that possible? In the slide, there is a network formed by enzymes that might be the decision center of the olfactory in the Drosophila. However, there are the experimental studies on these mechanisms are still lacking, but they're emerging, and we're starting to know the connectome and how the connectivities between those enzyme networks. So another of my future direction is to look at this network and see how we can make a robust decision depends on the different contexts. And the third direction is that as mighty as the Drosophila is still is not a very complicated organism. So I want to move from Drosophila to more complicated biological mechanisms like mammals, including humans. And there are differences in this. Even though the structure of olfactory circuit is relatively conserved, there are differences between the circuit in insects and mammals. For example, there are no adult neurogenesis in the insect brain, but there are in our brain, in the brain of mammals. And also, there are only one type of projection neuron in the insects, but there are two in mammals and in fact in all land vertebrates. And what this suggests is that evolution might evolve a parallel pathway to transform older information. And why do we need two parallel pathways to do that? And what's the usage of the newly evolved pathway is another interesting thing to look at. And then the last part is the structure of the olfactory circuit resembles some other brain regions, such as the cerebellum. So it could be interesting to transfer what we have learned in olfactory circuit to other brain regions. So with that, I'd like to thank my advisor and our collaborators, and thank you for your time and attention. Is there Or is there any questions? Yeah.
Can you talk a little more about the difference between how you implemented, the synaptic scaling versus batch normalization?
Oh.
Like the algorithmic detail differences between the two approaches?
The synaptic scaling is quite simple actually. I don't have that here. So the batch norm, you first standardize it, and you add some scales or new means to the data. So for the synaptic scaling, what I did is I just subtracted the mean without the standardization, and I led it to multiplied. So standard first, and then I have it multiplied by a learnable parameter. So basically, it scales the synaptic strength, but not standardized it. So does that make sense? So it's a very simple model, and it resembles the batch normalization, but has some because the division might cause some problems as well, especially because then division is small. Even if you add another small value to avoid division by 0.
Are there any machine learning tasks where you think synaptic scaling would be a better approach than batch normalization?
We haven't tested that. So for classification, so by other tests, do you mean are different data sets or different paradigms, like classification versus regularization for
I just mean, so batch normalization is kind of using the field, we're using this alternative approach. I was wondering if there's any field of people doing some sort of machine learning task that you're like, you guys should be doing it differently.
Depends on the data as well, and for example, in RNNs, you cannot use batch normalization because you do not take this in a batch, right? So that's why people have come up with layer normalization, where you can do normalization with a layer instead of a batch of inputs, and for synaptic scaling, I think it can be modulated to do a little bit both like that, and I think it works best also in a scenario with a batch of inputs, just like batch normalization, and so basically it's just a test, a proof of principle here to show that actually a very similar and a very simple normalization inspired by the brain can also do the similar things as batch normalization. So we're not intended to say that batch normalization actually much better in any sense, but so I think it's more important to notice that similar things happen in the brain, and we can take more inspirations from the brain to help develop new algorithms or new normalization algorithms. Yeah, does that answer your question?
But the brain is, well, I think it's hard to think about the normalization, because normally you think that the amplitude of the pulse is proportional to the impression of the sensors, and then if you normalize this one, they lose this kind of information. I don't know, that's probably you see it in different space that, okay, I translate this one a probability space, and then we're talking different, but not directly to the brain, right?
Actually do a very good job by normalizing the data and preserving the information at the same time. So for example, in the olfactory system, you want to basically convert or shorten the activities of the neuron so that you don't get like overactivating in the downstream. So there's like device normalization that happens to help you confine the activities, but also it preserves the information of the data and with other mechanisms with other like local normalizations as well. So I think the brain actually goes a long way to make sure the data is well normalized at the same time preserve all useful information you will need in the future or for further analysis. So,
Yeah, because you need to consider you use different kinds of neurons. For example, when you're smelling, one is the sensors, or the one is the processing that happens inside of the brain, right?
yeah.
And then we're just talking about the normalization, and you can talking about probably is about all the habitation that you talked before.
Well,
is,
the point of like this homeostatic plasticity is that it kind of happens kind of everywhere, it looks like. And probably we're most familiar with it.
inside of the brain.
But I think we think it's happening everywhere.
Yeah, it is a very like universal phenomenon.
In the brain. Yeah, but in the sense of the sensor that you detect when you smell is here, is a molecular reaction that happened, right? And after that, only is action potential that is the propagation inside of the brain. But you don't know exactly, if you detect one action potential, you don't know exactly what is the information, right?
Yeah, you can say that. I mean, is what we perceive really what happens in the world? We don't know that. So we only know the action potential that extends to our like cognitive center. So yeah, but there anything.
You told 3 great stories of connecting like basic neuroscience insights to the machine learning, either new algorithms or like other algorithms or like not good at like building good algorithms. Are there any like neuroscience insights that you started with, you tried to build something out and it kind of failed? Or are you batting 1,000, you know?
Oh, I have a lot of examples. On the top of my head, I will give you this example about DPM, another large neuron in the fly brain, similar to APL. So only one this type of neuron in each hemisphere in the fly brain. So you would want like APL has a very simple and straightforward function to inhibit cancer cells. So it's then strong inhibition and perform the sparse coding. But you would like think there might be some like simple functional things for DPM as well, because there's only one neuron and it should only do one simple task. But actually it seems like it involves in a lot of things. For example, it regulates sleep, it consolidates memory, and and it also uses not just only one neurotransmitter, it uses I think 2 different neurotransmitters. So like I have spent a lot of time trying to figure out what DPM does, but I cannot. And I hope one day like when there are more experimental insights, I can go back and do that. But you have another question?
Oh yeah, I was just curious, you mentioned habituation, the Kenyan cell representations will change. Is there evidence for that?
Oh, you mean like do our perceptions of order change?
No. Do you know if like anybody has like, yeah, Kenyan cells across time and seen that they change?
I'm not aware of any experimental studies in the fly directly, but I don't know. I'm just going to pure speculation here. So that might be one of the reasons we see repositional drifts in here, and it might account for some part of that. And I think we do kind of experience that type of phenomenon in our daily life. So if you're like go to a perfume store and you smell like all those programs for a long time, and then come out. The smells the smell of very like our daily life objects will kind of smell differently before you go in there. And after a while, you will like smell them in the usual way you do, but you do kind of get that change of perception of the odors after a strong concentration.
Yes, that's right. But you also experience the other part too.
Yeah.
Sometimes the smell kind of remains.
Yeah.
Yeah, but you're right, yeah.
Okay. So is there?
Take this going.
Yeah, anybody on Zoom want to ask a question? I'm gonna stop recording, but please feel free to ask the questions.